Hello, welcome back for one last video in Chapter 9 of Intro to Biotechnology. So we've already covered the basics and principles behind how chromatography works. So in this video, we're going to discuss all the different ways that we can use column chromatography to separate our protein of interest from all of those other proteins that we do not want by taking advantage of a protein's physical properties. So in this picture, you're looking at just what a basic setup for column chromatography looks like. So basically, you've got a big tube that has a couple of different parts to it. So this tube can either be made of plastic or glass. It doesn't really matter too much. So the most important part of the column is our binding resin. So in this case, the binding resin is represented as a big column of beads. So we've already talked about this, but the whole principle of how chromatography works is that our sample that contains our proteins is going to be laid on top of this resin, and our sample is going to move through the resin, and what we're hoping for is we're hoping that our protein of interest will interact with these beads, this resin, in a way that most other proteins do not. With the idea being that all the proteins that we don't want pass through the column pretty quickly, and then our protein that we do want should interact with this column in some particular way that we can collect it later. So column chromatography is going to be highly effective at removing a high population of proteins that we do not want while retaining our protein that we do want. So like I said, the first thing that you're going to do is pack your resin into the column, and then once the resin has settled, you're going to layer your sample that contains all your different proteins on top of it, and then gravity is going to pull your sample down through the resin, and then you end up with the same principles that we talked about with regard to mobile phase and stationary phase. So the column has a narrow opening at the bottom that allows buffer and protein sample to pass through. So the idea here is that we are going to collect fractions, which is just, as the name suggests, a fraction of the total volume of the sample. We're going to collect the sample that passes through the column in small little volumes with the idea that our protein of interest should be in a much more pure form in one or two or maybe even three of those fractions. So we can get most of the proteins that we don't want in some of the fractions, and then our protein that we do want in some of the other fractions. So what we're going to see is that depending on what type of column chromatography you're going to do, your proteins may need to be in a particular kind of buffer. So let's say that when we did the lysis and we collected our crude protein sample, let's say that that buffer is not the right kind of buffer that we need for the next type of chromatography that we're going to use. So we need to change the buffer that our proteins are in. And the way we're going to do this is by using dialysis. So in this picture in the top left, you can see a sample being loaded into a dialysis membrane, and then once it's loaded in, the top is going to be tied off in the same way the bottom is. So the way this works is that dialysis membrane has very small pores that will allow water and salts to pass through, the salts being the buffer molecules, while much larger molecules like proteins cannot pass through. So in the picture in the bottom right, you can see inside the bag, we've got our protein sample that is in buffer number 1, the buffer that we don't need anymore, and we place the dialysis bag in a beaker that contains a lot of buffer number 2. So the way this is going to work is that the buffer molecules in the buffer that we do not want are going to be drawn out of the bag, and the buffer molecules that we do want are going to be drawn into the bag. But all the while, the protein is going to stay inside the bag because those protein molecules are much too large to diffuse out of the bag through those tiny microscopic pores. So doing this several times, continually taking the bag and putting it in a fresh beaker of buffer number 2 will eventually get us to the point where our protein solution inside the bag is submerged in nothing but buffer number 2. And then from there, we can open up the bag, collect our protein sample that is now in the correct buffer, and we can move on to our next application. So the reason why we want to talk about this is because, like I said, different types of column chromatography are going to require different kinds of buffer, and that's something that you will always need to be aware of, because if your protein is not in the correct buffer, it may not interact with the column, with the resin, in the way that you want or that you expect, and that can really derail your attempt at purifying your protein. So one of the things that we're going to want to do once we have done a couple of rounds of column chromatography, we've collected all of our fractions that we want, first thing we want to do is make sure that we know which fractions actually contain protein. It would be a shame to move forward with a fraction that doesn't actually have any protein whatsoever in it. So we've talked about a couple of different ways that we can do this in previous chapters. We can use spectrophotometry, whether it's doing a quick Bradford assay to check for the presence of protein. We can check the absorbance at 280 nanometers to check for the presence of protein. But regardless, we want to be reasonably certain that our protein of interest, in addition to just any proteins at all, are in the fractions to begin with. So if we want to know which fractions actually contain our protein of interest, doing a Bradford assay or checking the A280 is probably not the way to go. That's just for checking whether protein is there, period. We want to know whether our protein of interest is actually there. So a good way of doing this is to run a little bit of the fractions on an SDS page gel. So this way we can actually confirm that some purification has occurred. So if you'll notice here, our original load, which for some reason only contains 2 proteins, in actuality it's going to contain a lot more proteins than that. So if this is our original load before we actually perform column chromatography, you can see that we have 2 different proteins in here, one that's about 60 kilodaltons and one that's at about maybe 20 or 25. But once we do column chromatography, you can see that we separate these 2 different proteins into 2 different fractions, the smaller one in fraction 5 and the larger one in fraction 6. So this should give you an idea of how you can be sure by using SDS Page that column chromatography has done what it's supposed to do. Okay, so now let's talk a little bit about the different types of column chromatography. The first one is called size exclusion chromatography, and sometimes it is also called gel filtration chromatography. But I like the term size exclusion chromatography because it makes it very clear what this step is doing. So this type of column chromatography is going to separate proteins based on their size or based on their molecular weight. So this is a really good first step in a purification procedure because a cell lysate is going to contain a lot of different proteins that really run the spectrum in terms of how big or how small they are. So we can use this as our first step to basically get all the big proteins or all the small proteins, depending on what we're looking for. So the resin in this case consists of many beads that have microscopic pores in them. These beads may be agarose, it may be polyacrylamide, it may be something else, but the beads are going to have these little pores in them that very small proteins can actually travel through. You can kind of think of them as little channels. Large proteins are not big enough to fit through these channels, so they're just going to go around the beads. So this is going to be a little bit different than what we've seen in the past with SDS Page and agarose gel electrophoresis, where we are used to the idea of small molecules running quickly and large molecules running slowly. Well, in this case, it is the exact opposite. Because the very small proteins are going to spend a lot of time working their way and winding through those channels, they are going to run through the column very slowly. Basically, it's like they're taking a whole bunch of back roads or dirt roads instead of just taking the highway. Larger proteins, because they're not going to waste any time trying to go through those pores, they're going to move very quickly through the column. So in this case, you can see our first fraction that we collect contains a bunch of larger proteins, whereas a later fraction contains some of the smaller ones. And you can actually see how this works in an SDS page gel that we could use to confirm the purification after the fractions are collected. So we can use this as a first step. If our protein of interest is a very large one, then we can run our crude protein sample and collect the earliest fractions with the idea that our protein is probably in those early fractions, and we can discard the later fractions that probably contain much smaller proteins that we are not interested in. Okay, so let's talk about the next one, which is called ion exchange chromatography. Instead of separating based on size, this time we are going to separate proteins based on their electrical charge at a given pH. So this is a conundrum that we've talked about before. We've said that some proteins are positively charged, some of them are negatively charged, and some of them are neutral. That's what made SDS page kind of a thing that we had to talk about. Well, it turns out that a protein's electrical charge has to do with two things: its isoelectric point, or Pi, and the pH at which the protein is maintained, which is going to be determined by what kind of buffer our protein is in. So hopefully you recall that proteins are made up of amino acids, and some amino acid side chains can either be basic or acidic. Of the ones that are basic or acidic, these side chains will contribute to the protein's isoelectric point. So for example, if the isoelectric point of a protein is 8.0, what that means is that if the protein is maintained at a pH of 8.0, then the protein will be perfectly electrically neutral. If the pH is less than 8.0, then the protein is going to be positively charged, and if the pH is greater than 8.0, the protein is going to be negatively charged. So if we're going to effectively use ion exchange chromatography, you need to know the isoelectric point of your protein of interest so that you'll have an idea of how it's going to interact with your column. So because we can separate proteins on the basis of whether they are negatively or positively charged, there are two different types of IEC that we can perform: cation exchange chromatography and anion exchange chromatography. So the resin is going to be made of beads, as you could probably predict, that either have a negative or a positive charge. Cation exchange chromatography beads will be negatively charged so that they can attract positively charged proteins, and the opposite is true for anion exchange chromatography. So for example, let's say that your protein of interest has an isoelectric point of 6.0, and it's in a buffer that has a pH of 8.0. So what this is going to mean is that your protein of interest is negatively charged. So you're going to want to perform anion exchange chromatography. So you would need to pick a binding resin that maintains a positive charge so it can interact with your negatively charged protein. So your negatively charged protein, along with all the other negatively charged proteins in your sample, are going to stick to the beads very tightly, while any neutral and positively charged proteins in your sample are just going to pass through the column very quickly. So if our protein of interest is negatively charged, we would want to collect it in a later fraction, because the first fractions are going to contain all the neutral and positively charged molecules that we do not want. And once again, you can, of course, confirm this with SDS page. So if your protein is negatively charged, it's going to be stuck to the column as long as the column is positively charged. So we can elute our protein off of the column and collect it in fractions, either by disrupting this attraction with a high salt concentrated solution, or by changing the pH of the column to get our protein to then become neutral so that it falls off the column. We can do it either way. Okay, the next type of chromatography is called affinity chromatography. This takes advantage of a protein's unique binding capabilities, meaning that some proteins have a high affinity for other types of proteins or other types of molecules. We've already seen how this works with antibodies. An antibody is a protein that has a high affinity for its antigen. So in the picture that you're looking at here, we are using beads that are coated with an antibody against our protein of interest. And if we pour our sample through the column, then only our protein of interest will bind to those antibodies and everything else will fall out of the column very quickly. So this method seems highly superior to other methods because the bead will literally only interact with our protein of interest and nothing else. But you're not always going to be able to use this type of chromatography because not every protein has some strong affinity that we can take advantage of. So you would either need to have an antibody against your protein of interest, which is not always the case, or your protein of interest has to naturally bind to something that we can stick onto a bead. Another example of affinity chromatography that is very common, if you end up working in a lab, you will probably work with this at some point, is that we can use recombinant protein technology to genetically engineer a histidine tag onto a protein. So histidine is one of the 20 amino acids that can make up a protein. So if we just add a coding sequence at the end of our insert, our gene that we stick into a plasmid that we're going to transform cells with, we can cause our protein to express an extra 6 histidines on the end of the protein, or at the start of the protein if you really want to. So the way that we're going to take advantage of this is that we can have beads that have nickel ions attached to them. Nickel has a high affinity for histidine, so if our protein is the only one in the sample that is tagged with histidine, then our protein should bind to the beads and everything else should pass through. So it's another example of affinity chromatography, a very common one at that. So again, we are taking advantage of a molecule binding to something with very high affinity and very high strength. And then once our protein is stuck to the column, we can elute it off with a chemical called imidazole, which is an analog of the side chain that makes up histidine. So if we look at this STS page gel, we can actually see very well that this affinity purification worked well. So in the leftmost lane, we can see the whole cell lysate. You can see that we actually do have quite a lot of our his-tagged recombinant protein, this big fat band right here. So after we pass our sample through the column, the flow through the buffer and protein that runs through the column without interacting, you can see here. And you can see that we have all the same proteins we had in the lysate except the protein that we were after. The protein that we are after shows up in the first elution fraction, and you can see that there are no other proteins there. So we can be confident at this point that we did a good job of purifying our protein of interest. And this sort of evidence is exactly what we are after at the end of a purification using chromatography. So one of the last types of chromatography that we're going to talk about, at least for column chromatography, is called hydrophobic interaction chromatography. So in this case, we are going to separate proteins based on their hydrophilicity or hydrophobicity. Basically, how much of an inclination do they have to interact or not interact with water. So the resin in this case is made of highly hydrophobic beads that repel water. So if our protein is also hydrophobic, it will like to stick to the beads. But if our protein is very hydrophilic and likes water, then it's going to be repelled by the beads. So what this is going to cause is that hydrophilic proteins will move through the column very quickly, and hydrophobic proteins will elute in much later fractions. So yet another physical property that we can take advantage of to separate proteins based on some aspect of them. So usually the case is going to be is that you can't really just get away with one type of chromatography other than affinity chromatography. So affinity chromatography should do a pretty good job of purifying just your protein of interest. But if you don't have affinity chromatography as a choice, then you're probably going to need several steps to achieve maximum purity. So this table that you're looking at is called a purification table, and it keeps track of the total amount of protein and your protein of interest as you go through the purification steps. So what you can see here is that the total activity, which is listed in this chart right here, indicates how much of your protein of interest is actually present in that step. Now, you might be alarmed that the total activity decreases as we go from step to step to step, but that is mostly because we are losing total protein at each step. So what we really want to pay attention to is the specific activity, which is basically how much protein do we have as a fraction of total protein. So as the purification goes on, a higher and higher and higher percentage of the total protein should be made up of our specific protein, which is why you see the specific activity go up. So as the specific activity rises, our protein becomes more and more pure. But you will also notice that the trade-away is that at each step, the yield goes down. So the yield starts at 100% and then goes down to 35%, which means that, unfortunately, in the process of purifying our protein, we lost 65% of our protein of interest, all for the sake of getting a little bit of it in pure form. So this is unavoidable and something that we just need to deal with. 100% yield is just not going to be possible. So the type of chromatography that we've been talking about is called open column chromatography. And it's very simple, very easy to use, but it is rather slow because it relies on the force of gravity to pull the sample down through the column. So again, fractions are collected in tubes like what you're seeing here that will be tested for protein activity later on, whether it's a Bradford assay or something else. So like I said, this type of chromatography is not terribly efficient. It's not very quick. So if you're working in a biotech setting and you're going to scale up your process, you may want to use open column chromatography just to get an idea of the parameters that you will use for a scaled up protocol later on. So in contrast to open column chromatography, something like what we see here with fast performance liquid chromatography utilizes a peristaltic pump to actually drive the sample through the stationary phase, not only so that we can collect our protein fractions in a more efficient and quicker way, but also you can see in this case that the fractions are run through a UV spectrophotometer before being loaded into fractions. That way, obviously, if we are exposing the fractions to light at 280 nanometers, we know right away which samples are going to have protein in it. So we don't have to bother with manually testing fractions either by spectrophotometry at 280 nanometers or by doing Bradford assays, which are even more time-consuming. So like we say, samples and buffer are driven through the column very quickly by a pump. The computer, most of these things that you see here are actually computerized, so you don't really have to do a whole lot of stuff in terms of labor. The flow rates can be automatically adjusted by way of computer to make sure that we're maximizing the interaction between the sample and both the mobile and the stationary phase so that we can get good separation. And then, as I said, a UV spectrophotometer can analyze the fractions as they come through for activity or protein presence. So that'll allow us to know right away which fractions contain protein or are protein of interest, and we can avoid wasting time on fractions that are not going to end up mattering. So something that's very similar is called high-performance liquid chromatography, or HPLC. It's very, very similar to FPLC in that you are going to drive your sample and a solvent through a solid column, but more often than not, HPLC is actually used for identifying particular molecules. So molecules can be identified using HPLC because molecules have their own unique retention times, or the amount of time they spend interacting with the column before they end up eluting off. However, everything else that you see here should be pretty similar to what we saw with FPLC. We detect our molecule of interest by using a UV spectrophotometer or some other way of detecting the molecule of interest. It is worth mentioning that HPLC doesn't have to be used for just detecting proteins. It can be used to detect a number of different molecules that are capable of being detected with some sort of method like spectrophotometry. So the last thing that we want to talk about to round out our discussion on column chromatography is the difference between equilibration and elution buffers. So we've already mentioned that different types of column chromatography, especially those involved in ion exchange and affinity chromatography, the types of buffer you're going to use are going to differ depending on the application. That's why we talked about the necessity of using dialysis to switch buffers when necessary. So equilibration buffer is basically just going to be used to set the stage just right for your protein to interact with the column. So this is especially true in ion exchange chromatography because we want to expose the resin to a buffer that has a pH that will promote binding of our protein to the resin. So if we're doing cation exchange chromatography, we want to make sure that our buffer is setting things up so that the resin will be negatively charged. If we pick a buffer that does not have the correct pH, then the resin may not end up being negatively charged, negatively charged. It may be positively charged or neutral instead. So we need to be very careful about what kind of buffer we are using in this case. So like I said, equilibration buffer is essentially here to promote the binding of our protein to the resin and nothing else. So it differs from elution buffer in that elution buffer usually has very harsh makeup to it. So elution buffer is going to discourage the binding of your protein to the column. So really what we're using elution buffer for is once we have a high degree of confidence that we have removed most of the contaminating proteins from the column and mostly what is left is our protein of interest, we can run some elution buffer through the column, and this should break the interaction between our protein and the column, and our protein should safely fall through the column, and we can collect it in a fraction. So elution buffer can be harsh in a number of different ways, whether it is high salt concentration, extreme pH conditions. We saw how imidazole is a chemical that is used in the elution of his-tagged proteins. So the type of elution buffer that you will use is going to differ depending on what type of chromatography you're using, just as what we saw with equilibration buffer. All right, so that is going to do it for Chapter 9. I certainly hope you found it interesting. Join us next time and we will get into Chapter 12 on medical biotechnologies. I look forward to seeing you then. See you later.